For today's lecture, we're going to get into some applications of fluorescence using diffusion, and then shifting over to other applications of light that use scattering. So either we'll talk about dynamic light scattering, which is using Rayleigh scattering, and then also Raman scattering, that's inelastic scattering. So we'll be covering, I guess, 3 different types of areas using light to look at dynamics. Okay. So, like I mentioned on the title slide, we're going to be talking about fluorescence to measure diffusion. And scattering as well. So we can get diffusion out. We can also get the number of particles out. We can get the particle size out. We can get the concentration out as well with these techniques, and they take advantage of fluctuations in solution that arise from the random diffusion in motion. And then we'll shift gears after halfway through. We'll talk about Raman scattering. And that has some vibrational information and can give chemical identification information and imaging and spectroscopy. And we're also it's important to know that it's different, how it's different from IR or fluorescence. And then we'll talk about some of the applications of Raman scattering. So while we'll be discussing, I guess, 3 different types of techniques, we're still in the light mode of this course where we're talking about photons. And we'll be mostly working in the visible range today. The samples will either be fluorophores for the fluorescence-based techniques, or large enough particles that scatter light, or something that has a unique vibration, that's Raman active. So these are for the 3 different methods we'll talk about today. And again, the detector, we're still detecting photons. It's going to be some type of photodetector. And it's either going to be from the emitted photons from fluorescence or scattered photons. From the source. So again, it's not going to be like the UV vis where those are the same photons. So jumping into some applications of fluorescence for measuring diffusion and size and number of particles, the technique we're going to talk about today is called fluorescence correlation spectroscopy. Or it's also called FCS. What you have in FCS is we're going to actually start talking a little bit about microscopy today. And I'll go over some details of the microscope in a couple of slides. But what you have is you have light focused through the objective of a microscope, and that focuses your light. Typically you think an objective is a very fancy, let's say the lens is facing this way. Sorry. My sketching is bad. You have light coming into a lens. We typically draw with ray optics that it's being focused to a single point here. At that point, it's actually not infinitesimally small. It's going to have some radius associated with it, or not. And that's what I'm showing here, is at that focus point of light, you're going to have a Gaussian beam profile. So light's coming from the bottom here. So we have some radius or diameter associated with it. And that radius is typically diffraction limited for visible light. That's 250 nanometers or so. And in fluorescence correlation spectroscopy, you have this focal volume of your light, and you have it focused over your sample. I have a cover slip. This is just a cover slip drawn here. And what you're measuring is you'll have fluorescent biomolecules. If you have GFP or you have a protein tagged with a fluorophore, just like we talked in last lecture, a DNA molecule tagged with synthetic fluorophores. And they're in solution and they're able to freely diffuse in the solution, or they can be in a cell. And when they diffuse and they pass through the focal volume, what you'll see on a one-dimensional detector, if you have intensity versus time here, if nothing's in the focal volume, you'll use a very low concentration here. But when a molecule enters, you'll see an increase in intensity. And when it leaves the focal volume, that'll decrease. And you'll have fluctuations here, increases in intensity from many molecules passing through that focal volume. So to have just isolated individual molecules, one at a time in the focal volume, you use low concentrations of nanomolar concentrations of fluorophores. And you want on average less than one molecule in your focal volume at a time. So you have these individual increases in intensity. From this intensity trajectory, this intensity versus time, you would then calculate the fluctuations in the intensity. So we're gonna write that as delta F of T. You would take the fluorescence intensity at a certain time and subtract the average fluorescence intensity. So this is the fluctuations in intensity. And from that, you'll perform autocorrelation of your signal, of the fluctuations in your signal. So I'll go over on the next slide just a reminder of what correlation is mathematically and give some examples. But we're gonna write this in FCS. Typically G is used to represent the autocorrelation. Tau is shifting your signal versus itself for a given lag time. So this is what we call the lag time. And we will take a fluctuation in intensity at a given time t and multiply that by the same fluctuation intensity shifted by that lag time tau. We take the average of that over for all of the time lags here, and then we're going to divide that by our average intensity over the entire trajectory here. So this term in the denominator normalizes your autocorrelation curve. The term in the top is your autocorrelation. So after taking this autocorrelation and performing this for many values of tau, your lag time here, you get your resulting autocorrelation curve, where it's going to be a maximum value of 1 since it's normalized at a lag of 0, because your signal is exactly the same as itself. But then as you shift it, you're looking at the self-similarity as you shift it versus time. And this will decay versus tau. And if something is small, it's going to decay faster than if you have something that's large. So this is large. This is small. And in the PDF of the notes, I have actual data from, let's say the small thing is going to be an rhodamine 6G, or like a small organic fluorophore like we talked about last lecture, while something large might be a nanoparticle, like let's say a 40 nanometer nanoparticle, that the difference will depend on the size. And you can think about this, if we're measuring intensity versus time, something large will diffuse slower. It's going to be in the focal volume longer. So if this is just one particle and you perform that autocorrelation and shift it over, it's going to be more self-similar for longer values of tau compared to if you have something small. If you shift it by the same amount, the overlap's going to be 0 there. So based on these autocorrelation curves, this guy. You can then do some curve fitting. To an analytical model that accounts for the size, the concentration of the particles. So for FCS, the curve you fit to, the autocorrelation curve, it's 1 over the effective focal volume times your concentration of your molecules. So this is the number of molecules. This entire first term is 1 over the number of molecules on average that are in your solution. This is then divided by, multiplied times 1 over 1 plus your lag time tau over tau d. Where tau d, this is your characteristic diffusion time. And this characteristic diffusion time is on average how long your molecule is staying within your focal volume when you see these increases in intensity. And then this is multiplied times 1 over the square root of 1 plus r naught squared over z naught squared. So this is the radius over the height of your focal volume. So this is the confocal focal volume here, plus, or sorry, multiplied, it should be multiplied, again, tau over tau d. So this is what you fit FCS data to. Square this guy off. This is exactly why I upload the PDFs, because it's much nicely organized compared to this. And this characteristic diffusion time you can then relate to the diffusion coefficient. So the diffusion coefficient would be r naught squared. So again, this is the radius of your focal volume times, here what I've shown here is 3-dimensional diffusion. So this would be 6 times tau d. But if you were looking at 2D diffusion, it would be 4. If you were looking at 1D diffusion, it would be 2. That's the degrees of freedom. You can imagine 1D diffusion could go left to the right. 2D diffusion, if you were measuring something in a membrane, you could go left, right, up and down, and in 3D, in and out as well. And so you can get the diffusion coefficient, but this diffusion coefficient then also relates to the size of your molecule with the Stokes-Einstein equation, where the hydrodynamic radius of your molecule is kBt over 6 pi times the viscosity of your solution that I'm representing with rho here, times the diffusion coefficient. So you can see there's a lot of variables here in the equations that I'm showing. But FCS can give you diffusion coefficient, it can give you the number of molecules that you're measuring, it can give you the concentration, it can give you the size. So these are all, depending on how you design your experiment, you can get these different values out.
Professor Kisley?
Yeah.
Sorry, could you remind me what RH is again?
So that's the hydrodynamic radius.
Okay.
So that's the, let me write it out. So that's the effective size or the radius of the particle or molecule that's diffusing. That's taking into account that it's in an aqueous solution and that there's going to be solvent molecules associated with it that will change how fast it's moving. So that's why there's also this term related to the viscosity in here. Because you'll see for different types of measurements, you might see like the radius of gyration. There's different ways that you can define what your radius is. So this is a specific one. Okay, any other questions? Okay. And since FCS, and then when we're talking about dynamic light scattering, These are all based on correlation, so I want to make sure that we're all up to speed of what I mean when I'm saying correlation. I'm talking about the mathematical function of correlation, not, you can also say correlation is if you have some data plots and you can say like, oh, there's a linear trend, there's a correlation between those. That's not what I'm talking about when I'm mentioning correlation. When I'm talking about correlation, I'm saying it's a way to measure and quantify the similarity between a signal and itself. Would be autocorrelation, which is what we're doing in FCS so far, or a signal and another signal, which would be cross-correlation. And we'll talk about how that's used in FCS as well. But the definition for correlation traditionally would be, and I'm talking about cross-correlation here with, that's why I'm saying XC, it would be the integral from negative infinity to infinity of your signal, one signal f of t, times your second signal h of t plus tau, integrated over all of time. So this is a traditional definition of correlation, and if f of t equals h of t, that's going to be autocorrelation then instead. What I showed for FCS, you can see that there are differences in that FCS, since we're doing a real physical measurement, we don't have any negative time values. So FCS only is going from 0 to the positive extent of your data. So it's only going to, let's say, T max, or maximum measurement. And then in FCS, we had that denominator term that's for normalization. So that's why there's some small differences. But what this means graphically, it's nice to show, if we again have our intensity versus time. If we just had a single square wave pulse, what this means is this tau value, maybe I'll try to play around with pen colors here. Is for one data point on, let's have our G of tau versus tau here, is what you would do is you would multiply these together, look at their overlap. That's what the multiplication is doing, and you would get a single value for this overlap. If we took, it's always at the maximum when you have a value of tau of 0, because you have that complete overlap. So you'll start at 1 here. And then as you shift your signal by larger and larger values of tau, that overlap gets less and less. And for a square wave, you're just going to get a straight line that you would see. So let's say if we have a much shorter pulse, how would that change your autocorrelation curve here? What difference would you see in your plot for your autocorrelation for a square wave pulse that's smaller than this red one that I show up here? So if I sketch this out, if I shift this over by smaller amount, you'll have less overlap. You're going to see a faster decay for a smaller square wave here. So that's why when you have something that's smaller, diffusing faster, you have shorter bursts in intensity than if you have something longer, so you see a faster decay with that. So that's just a refresher. So FCS is using, I mentioned a confocal microscope. We're going to go into microscopes in more detail next Thursday. But to walk you through what I mean by a confocal microscope, you typically have a laser light source. You're going to pass that excitation source to an objective on a microscope, where this dichroic filter is a special type of mirror that will reflect shorter wavelengths and allow longer wavelengths to pass through. So you're going to overfill. The important thing with confocal microscopes is you overfill the back objective of the objective here. So then you tightly focus your laser light to a single point. That's diffraction limited here with that 250 nanometers. So that's what this light blue is representing here. So with confocal microscopes, you're going to have a point excitation source that's diffraction limited, about 250 nanometers in diameter, about a micron in height. So then the intensity from those fluorophores entering the volume will pass back through the objective, pass through your dichroic. That's what the green light is representing here. And you then focus it on a detector. And typically, confocal microscopes have a pinhole, and this was a key development. That if you have a pinhole present, you can block out of focus light. If you focus your emission light through this pinhole, you can remove any unwanted signal from out of focus fluorophores. And then the detector here is typically a 1D detector. It's not going to be a camera that collects an entire image. It's just going to be intensity versus time here. Where usually these are APDs or avalanche photodiodes. So these are very sensitive to detecting individual photons. And you're able to collect down to typically microseconds to minute-long timescales for your correlation. So you're collecting data over 10 to the 8, I guess, decades of time when you're collecting FCS data here. These cartoons show over here, if you want to try to do some imaging with a confocal microscope, you have to scan over that. And we'll go into more detail about that next week when we talk more about using microscopes for imaging. Here you're just sitting at one single point. I also want to mention that FCS was developed in 1974 by Elson and Madge out at UC San Diego. And so it's been around for quite a while. And traditional FCS is at one point. It uses one color, and you really need fast fluctuations. If you're trying to monitor anything that's slower than, let's say, like 10 seconds or so, or single seconds, you'll run into some issues of not having enough intensity fluctuations to produce a correlation curve in your data. So since 1974, people have come up with lots of variations on FCS to try to overcome some of its limitations. So traditional FCS is limited to that one point of around 250 nanometers. If you want to beat this diffraction limit of light, it's called super resolution, which we'll talk about in more detail later. But I wanted to show one example here where you can see this is doing FCS in a membrane environment. You can see with the traditional confocal, you have this large spot that they have about 4 fluorophores in their little cartoon here. There's a super resolution technique called stimulated emission depletion, or STED. We'll talk about that more later. But what they do is they have this donut shaped, if you looked at the top down, this would be the red and this would be the green. You have a donut shaped laser beam that makes any of the molecules in the red donut dark, and only things in the center would be bright. So you can get down to tens of nanometers instead of hundreds of nanometers. So they've incorporated super resolution. We have a super resolution STED confocal microscope in the imaging core in School of Medicine. So we'll talk about that in like that's going to be like at the end of the month, we'll do another quote visit over there, but there is a STED microscope that can do FCS with these small resolutions. So that's if you're looking for small features. People have also worked with 2D FCS, so working with images instead of just a single point. I mentioned that there's cross-correlation in addition to autocorrelation, and this uses 2 focal volumes, which I've shown here. They have a blue and a reddish colored focal volumes. They're going to be some distance apart from one another, and you can track 2 fluorophores instead of just 1 to get intensity fluctuations from your 2 channels, and you perform cross-correlation instead of autocorrelation. And this can tell you things about like association between the same 2 different colors. So let's say you have some binding partners. If you see that their cross-correlation, excuse me, is high, that would mean that they're both in the same location at the same time. So here, I guess these focal volumes are overlapping. You can also have them separated by a distance and do cross-correlation to get distance information. If you're trying to look at something that's slow, there's scanning FCS, where you move your focal volume over time, like the location changes. So you can look at things that are moving slowly. And then there's also, you can do FRAP, which we discussed last week, and multicolor FCS if you're using multiple labels. So you can look at the conformation with that donor acceptor ratios, or if you remember, the efficiency is the intensity of the acceptor over the intensity of the donor plus the acceptor. Will tell you about the distance between those 2. So you can look at the conformation in addition to the diffusion. So there's many, many different variations on FCS. If you guys want to learn more about FCS, I'll point you to Petra Schwilla, who's out, I think she's at Max Planck Institute in Germany, but she has a lot of great reviews going over FCS, mostly in the 2000s. Now she's moved towards looking at bacteria more, but it is a powerful technique that you can see. I also want to point out, I said that we have a STED FCS in the imaging core. We also have a lot of, there's I think 4 confocal microscopes that are over in the microscopy core. So you can get commercial setups that kind of have a black box, like click a button and it's going to spit out what your diffusion coefficient is or what your concentration is. But home built setups will give you like that intensity versus time trace where you can manipulate it on your own. Commercial instruments don't always have that. But most confocals will have an option of having FCS, or it's worth asking. I know there's several on campus. I also want to mention an additional technique that uses fluorescence to look at diffusion called FRAP. That's fluorescence recovery after photobleaching. And the concept behind this is kind of straightforward. You can see here is an image of a cell, and they focus on one little area, and you scan your confocal focal volume over this area over time. And you use a very high power on the order of a couple milliseconds to bleach. So this is before, this is the pre area, and this plot here is the intensity within this red circle. So you use a high laser power at one point in time to bleach the area, and then the rearrangement of molecules will then diffuse back into the bleach spot. And you look at the recovery over time, and that's that curve here. And you fit that curve to an empirical equation where you look at your intensity versus time compared to the initial intensity, and you just fit with an amplitude and a rise time and a stretching factor here. And this tau d, you can then again relate to the diffusion coefficients. And this is some data that I took of looking at proteins in a hydrogel environment and looking at the recovery over time. So fluorescence recovery after photobleaching is an alternative to getting the diffusion coefficient. You can see with the equation, it's not as detailed, it's not as specific as the FCS equation. And you're limited to, I'd say, tens to hundreds of microns versus FCS is going to be 250 nanometers for your spatial resolution. So you can't use this to look at very small areas. People use this to look at, let's say, ex situ or in vitro samples, or things taking place in the membrane. But it does come up quite a bit and is pretty available. So you'll see this in biophysical papers. Okay. So those are some, since we talked all about fluorescence last week, here's two examples of, or two specific fluorescent methods that are used to extract dynamic information about the diffusion and number of particles. Similar to FCS, but using a different phenomenon of light is dynamic light scattering, or it's abbreviated DLS. And it's similar to FCS, but uses scattering instead of fluorescence. So here we're talking about elastic scattering. It's also called Rayleigh scattering, since Lord Rayleigh defined it. And with elastic scattering, what this arises from is your electric field of your light causes oscillations in the electric cloud of your molecule or your particle. So they'll be oscillating at the same frequency. And then your molecule or your particle becomes a radiating dipole. So your incident light wavelength is the same as your scattering wavelength. And the energy of your electric field or your light here isn't enough to put your molecule in an excited electronic state. So that's how it's different from fluorescence. Fluorescence, you're going to an excited electronic state. With scattering, you're not in one of those excited states like fluorescence. You can see here that the type of scattering, me scattering, is a type of more specialized area of Rayleigh scattering. But depending on the particle size, that will change the directionality of the scattering of the light here. But DLS can measure particles in all of these regime sizes here. You can see that these green arrows here are just showing that with me scattering, you're gonna have more forward scattering than with Rayleigh scattering. That's gonna be more isotropic, shown here. But with Rayleigh scattering, I have the equation written out here of the intensity of the scattering. It's going to be dependent on your incident intensity here, the refractive indices, the angle that you're observing it at, the distance that you're observing it from. But the important parameter that I want to point out here is that it depends on the size of your particle to the 6th value, or to the 6th power here. So depending on the size of your molecular particle, like you'll have more, it greatly changes as your particle increases. So DLS, the particle has to be large enough to scatter enough light significantly. So it's important if you're using DLS to know the limits of your instrumentation. With the setup of a DLS, these are typically more black box type instruments. You'll see Zeta Sizer or Wyatt are common vendors for those. DLS is taking account of the same fact of, okay, you have some laser light source. You're gonna then collect some intensity versus time of the scattered light, and you're gonna see these fluctuations based on the particle size and its diffusion. Since you only wanna detect the scattered light, oftentimes you have this 90-degree setup, or more recently, you can have something where your detector will be 170 degrees. And you can see with this term in Rayleigh scattering that it's to the cosine squared of the angle. So if you're working at 170 degrees versus 90 degrees, you're gonna have a higher signal. If you're working slightly off axis. You're not gonna work at 180 degrees because that would then collect all the transmitted light as well. And on campus here, okay, here's a question. Andy's asking if this is derived from the Clausius-Rosati relation, and I'm not familiar with that relation, so I'll have to look that up. I don't know if you want to expand on it.
The n squared minus 1 over n squared plus 2 is the left side of it.
Okay.
So do you know how it's derived?
Off the top of my head, no. I'll have to I'll look into that and get back to you.
Right, thank you.
Yeah. That's a quick answer to that. Okay. Yeah, and I don't know if with the equipment on campus, I know when I've used DLS, I found it more in polymer departments and material science departments. I don't know if the two or three people in the class from macro know if you guys have a DLS in your department. But it's less commonly found. I would say I don't see this in biophysics facilities, but it can still be used for biophysical applications. Okay. So that's why I want to, okay, let's see what the chat says. Yeah, okay. So Catherine's saying that there's one in her own lab. So yeah, because looking at the globule states of polymers, that's why you find it more often there. Okay. So we've talked so far about 3 different techniques that you can get diffusion coefficients out of. Also with the DLS that I have here, DLS, the black box aspect of it, they don't typically give you your intensity trajectory versus time. You usually have to put inputs of refractive indices or the viscosity, and it'll spit out the particle size or whatever value, or the hydrodynamic radius that you're looking for. But it does use math that's slightly different from the FCS equations, but it effectively does something similar where you're going intensity and autocorrelation to diffusion coefficient or size or n and the like. But I didn't want to go into a separate format of mathematical equations for them. So since we've talked about FRAP, we've talked about FCS, and we've talked about DLS, I wanted to do a breakout question of, let's say your PI comes up to you and says, I want you to do this type of measurement, or I want you to determine this value, looking at the size change in a gold nanoparticle. I have 5 different descriptions here. I'm going to split you guys into random groups, and if you can look at your problem, I'll give you guys 10 minutes or so to discuss it, and then if you guys can pick again one person to report back and describe which of the methods would you pick to use to figure this out, what would you be trying to measure, and why would you pick that method versus the other ones? Why wouldn't the other methods work? So I'm going to try to do breakout groups. Breakout rooms. So I've randomly put you guys into groups. I'll stay in this main area. If you guys have questions, please ask me here, and then let's meet back at 12:30 or so, and you guys can discuss and represent. And if you guys finish early, take time to read the other descriptions and discuss some of those. Sound good? Any questions? Okay, I'm gonna open all the rooms then, and hopefully you guys will be putting them on. Okay.
Oh, okay. Hello. None of us had the problems recorded, so then
Okay, screen grab.
yeah. That was a mistake. Oh. Oh, I can't.
Okay. 'Cause, yeah, I think we can do that. So, yeah, we'll just do the FRAP. So, FRAP is 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 FRAP So is everybody out of the rooms now? Okay. Cool. So someone from group 1 wants to talk about, I guess, their situation, what method they picked and why.
Okay, so I'll go ahead. I'm from group 1. So our approach was first to say which one does not really work. So we thought the FRAP do not work because of the size of the particle. And this is like more like a larger particles. So we said that does not work. So we were talking about FCS and DLS. And I think we decided on DLS with me scattering because this is where the particle size falls in the between 50 and 500 nanometer. And yeah, we're like more measuring the scattering versus the excitation of that particle.
Yeah, so
is that correct? This is what.
Yeah, so I'd say correct in removing FRAP. And yeah, I would go straight, I would think, okay, it's colloidal, I would say DLS, but you could use either. So this one, either DLS or FCS would work, because you would have to know though, with FCS, you have to think, okay, something needs to be fluorescent, but you would have to know that these plasmonic particles have luminescence themselves, and it's not technically fluorescence, but you could use FCS. So either of those techniques would work.
Yeah, this is what was at the end, because you said in that it's photothermal, and that might cause excitation, and that might cause also for us to use FCS. So we're debating between the two, but we decided on the DLS.
Yeah, so like with the FCS, you'd have to look at the luminescence of the particle, and it would probably be more in the visible than the IR, but either of those methods would work. And then I would just say, for what would you be measuring? So DLS, you wouldn't be changing the wavelength. You wouldn't be looking at anything spectral. What you would be looking at is, you look at the intensity of that scattering versus time, and you would be comparing the particles alone and the particles with after being exposed to blood, and looking at the size difference. So
okay,
yeah,
great, thank you.
good. Okay, so let's move on to group 2.
So we chose FCS for this because the other ones wouldn't work so well, because DLS doesn't take advantage of the fluorescent tagging, and FRAP was over a larger area. So hopefully we'd be using FCS to measure the diffusion constant and determine if it's in liquid, gel, or solid based on that.
Yeah, so a challenge with FCS is that you have to have a low number of fluorescent proteins to be able to see a fluctuation. So here, if you wanted to try to use FCS, you would have to have the protein without the label and with the label, and I think that could work. And also, if it's in the solid form or gel form, it might be too slow to see fluctuations. So I think your logic of picking FCS because there's something fluorescence there is good. But traditionally, more in these type of experiments, people would just use, they would use FRAP. So you'd have like this I try to put some figures here to help illustrate it. So you see this big blob that's microns in size. I guess you'd have to bleach a little bit smaller of an area. Maybe this was a little misleading. But people traditionally use FRAP more for these details. But I think if you design it correctly, you could also use FCS, but it might be a little more challenging. That sounds good.
Right. And another question, we were wondering about the concentration, because you had said nanomolar. Is that a strict limit, or is it just a recommended, you know?
Yeah, so that's where in this description right here, since it's micromolar, that would become a problem. for FCS. If all the if all the protein was labeled in fluorescence and micromolar concentrations, and that's all you had there, you wouldn't see those fluctuations. You would see a constant amount of fluorescence and maybe some small, it's hard to tell some changes there if everything's fluorescent. So yeah.
Great, thank you.
Cool. Okay. Let's move on to group 3 then.
Hi, I'm presenting for group 3. So our understanding of lipid rafts are just that they're like locations in the membrane with lower like membrane fluidity, so that maybe they would be moving in an aggregate sort of whole and possibly wouldn't be, it might be moving in a different manner than the surroundings. So we were looking at the different techniques, and we decided that DLS wouldn't really make sense in this situation, just because we wouldn't be really looking at scattering because we have the fluorescent tags on the GM1. So we were debating between FRAP and FCS, and we thought that FRAP might be appropriate just because we're looking over a large like area of like the plasma membrane, I thought. And then we were also looking at FCS with the 2D FCS because we thought that could also be a potential way of looking at, I guess like membrane. So I mean, we were kind of debating between the two, and we weren't sure exactly which one would be more appropriate.
Yeah, so I guess the hint in this description is that how large would you expect like these lipid raft or these different domains to be? So since I put in there nano or micro domains, which technique do you think could get down to those scales?
FCS.
Yeah, so for this one, FCS would be a better technique to pick if you want to get down to that smaller organization there. And FCS is used quite a bit in looking at dynamics in membranes. Okay.
I think this would be 2D FCS, right?
Yeah, it would be 2D FCS. Exactly.
I just like a really quick sort of question. So like because like so we were when I was like looking at this, I was thinking that like we would be looking at a very large like region of the plasma membrane, and then we'd just be trying to see if there were like dynamic changes in the how the GM1 like fluorescent tag would be moving in comparison to the surroundings. So like we would be, so but like in this case we would actually be looking at individual nano and micro domains of the lipid rafts. So like we would want to go into that length scale kind of.
Yeah, so if you bleached a large area in FRAP, 10 microns or so, you're going to have quite a bit of GM1, and you would have areas that are ordered and disordered in that large of an area. So if you really want to isolate those two different areas, FCS could actually start to resolve those. You might be able to see it in FRAP with seeing like two curves in the recovery, possibly two components in the recovery, but it would be more challenging with that resolution.
Okay, thank you.
Okay, so let's move on to group 4.
Yeah, so we weren't really sure. The first thing I thought of is just like FRET, since it has a fluorophore quencher, but that's not an option. And then also like on the first homework, like circular dichrosum spectroscopy would work, but that's also not an option. So DLS we eliminated since you're not taking advantage of fluorescence at all, and there seemed to be a lot of emphasis on the fact that it was fluorescently labeled. FRAP like just doesn't have the spatial resolution. But then even super resolved FCS, like tens of nanometers you can do, but not like angstroms to look at the secondary structure of DNA. So we just kind of looked around in literature and saw what like people were using, and it seemed like of those three, FCS was used the most often, but still used very rarely. Yeah, honestly not too sure on which one.
Yeah, so this one was probably the hardest one. So thinking about, yeah, there is some FRET aspect here, except it's with a quencher instead of an acceptor. So with FCS, with this conformational change, you could see a change in the diffusion coefficient possibly, but you'd also see a change in the brightness here. So this does have aspects of FRET. But yeah, FCS would be the choice here, but one of those more advanced methods of FCS, where you're going to have an intensity dependence based on the conformation of the DNA in addition to the diffusion coefficient. So this was the hardest one.
So if there's like a way you could, or I guess you know the relation between like the secondary structure and like I guess, the conformations it takes in the way the particle or the DNA diffuses. And that's why you'd use
Here-
FCS, or am I?
So here you would see lower intensity versus if it's extended.
Oh, versus.
Changes in your FCS curve based on
Oh, okay.
magnitude on the y-axis, but then also the curve shape possibly if this is large enough that if it's random, it should diffuse slower than if it's base paired up. Good job with going through the literature and stuff to find some hints there. And then let's move on finally to group 5.
So we were primarily sort of deciding between FCS and DLS. And I think we settled on FCS primarily because of the fact that typical monoclonal antibodies are in around like the tens of nanometers range. So we sort of figured that FCS would be most ideal for that given the size, even with aggregation.
Yeah, so the only challenge here, so FCS, I think that's a good point about the size regime of where DLS could work with. I think individual antibodies, you'd be pushing the limits, so FCS may be more useful there, but you would have to be able to fluorescently label them. So if you just want to take these therapeutics as they are without putting a tag in, then DLS might be a better choice. Yeah, you'd have to know the specs of your DLS. Can it get down to tens of nanometers? I think with the aggregate size, you definitely should be able to resolve that. But with the individual antibodies, it'll depend on how good your DLS is. But it would be similar.
Marshmallow bar. Marshmallow bar. One with chocolate chips on it.
I think it's just background noise now. So with that, and in the interest of time, I think we're going to stop here and I'll hold off on talking about ramen until next Thursday. And in the meantime, if you guys have any questions about Tuesday's breakout session, or yeah, if you have questions about that, or if you had any issues uploading the homework or the problem sets, please let me know. But with that, I think on Tuesday, I'll see some of you guys outside, and then I'll create breakout groups in the same Zoom meeting for the other 2 groups that are going to be meeting online. So yeah, thanks for lecture, and I'll see you guys on Tuesday then, unless there's any other questions.